Thank you all for coming. There are seats up front. And I want to thank those who invited me to give this talk, which I cannot give alone as indicated on this slide. So I want to acknowledge at least some of the people who have helped educate me so that I can give this talk. And in particular with regards to virology, which is not my training, I want to thank Barry Bloomberg for making it possible to think about viruses in an astrobiological context. I want to thank John Barris, who might be known for other things, but his dissertation research was on bacteriophage and genetic exchange in the cold marine environment, which I found inspirational. Lead Wells was a former graduate student of mine who taught me more about viruses than anybody and who pushed a lot of lower temperature and salt limits for viral production. Matt Sullivan is a current collaborator who keeps me state-of-the-art. I helped to launch the UW Astrobiology program. I sit in oceanography, but what grabbed my attention the most about this field was that the sites we have to explore for life are all very cold. And I thought perhaps I could contribute to this discussion. My lab could contribute through the study of how microbial life manages in cold ice. Today we know, or we can think of the solar system as overflowing with liquid water because we now know of multiple oceans in the solar system. And so following the NAI mantra, I have been following the water for most of my career, even before NAI. You see a temperature scale here. Today I am, or in the last 20 years, I have been working on the lower end of the temperature scale where salt keeps water from freezing at lower temperatures. I've also been listening to the Inuit in the north where I work, who I believe are the original astrobiologists who understand that ice is actually the natural state of water in our solar system. So in fact, I've been following the ice and trying hard at that since we're losing it on this planet. So I tried very hard always to get funding to go into the Arctic in the dead of winter, which made me a lonely character with a few other people helping me. But one of the reasons for this is that saline ice can get very cold and still have a liquid habitable environment within it. So these are a series of panels. I'm going to show you a little primer on sea ice if you haven't thought about it very much. 30 millimeter panels here. The black area that you see is the liquid fraction of the ice. With 20% porosity. So 20% of this ice is liquid habitable space, and an average pore is over 2 millimeters in size. So this is a water world for microbes. But you just have to chill the ice down a little bit before you begin to narrow up these spaces. And when you get to wintertime ice, you close up considerably. And in the process, concentrate the salt that was present in the source waters. So at minus 30 degrees, At minus 30 degrees, you have very small pore space, 2% liquid volume in here, 24% salt in those brines, conditions that would normally we would think would shut down life. I'm here to tell you that is not the case. But one important factor here, or the important factor here, is the brine concentrating factor. Whatever salts were in the source fluid get concentrated in these small spaces as a function of temperature. And anything else that was in that source water also gets concentrated. To demonstrate this concentration effect, which is temperature driven, I'm going to show you the temperature range across sea ice. So here is an ice core that has just been pulled out onto the surface of the ice. And the next slide shows you an infrared camera image of this so you can actually see the temperature gradient. So here's the very cold ice at the top that was exposed to the atmosphere, and the warm ice at the bottom that's bathed by ocean water. So sea ice functions as a wonderful natural laboratory for all sorts of gradients. In the Cockle lab, it would be multiple extremes all at once. So here's the temperature range, getting to the eutectic of seawater where all of the salt precipitates out and you no longer have liquid. The salinity range here goes to very high until all the salts are precipitated out. The liquid volume fraction reduces to 0 under those extreme conditions. But most importantly for this talk is that the colder the ice, the higher the contact rate between particles or anything that's concentrated in those pockets. So that includes microbial life and viral life, or viruses, depending on your definition. So generally, in this gradient here, you can think about microbial growth easily happening under these conditions, then reducing to metabolism, and finally the strategies have to be about surviving. And enzymes and quorum signaling signals, like we just heard about, are involved in all of this. At these high contact rates, we have lots of free DNA and phage, and this is where I'll focus coming along. Exopolymers are central to the whole story. I'll talk a bit more about that. Please go see Max Showalter's poster this evening. He can tell you a lot more about this side of the story. And Zach Cooper in a poster tonight can tell you all about the hosts that we are finding in these extreme environments. The first thing we had to do was to figure out how to look into unfrozen sea ice. So what you're seeing here with no stain is a brine pocket. This is solid ice crystal matrix around it. You can see a few diatoms here. With a DNA stain, you can see all of the bacteria that are concentrated in there. With another stain, you can see these exopolymers, as we call them. They're polysaccharides that function as antifreeze for these organisms. And you don't even need a stain to see the goopiness of them because they fill every pore in sea ice. You can grow artificial sea ice from seawater with and without this exopolymeric material, and you get a completely different physical structure. You get Euclidean pores here, which are disconnected from each other. You get fractal pores here that are connected, very important for the life living in there. And you can increase the porosity. You can increase the habitable space of this ice in the presence of these organic polymers. So to emphasize to you that these little pore spaces in here under extreme conditions are not dead spaces. Here is our model organism happily swimming away at -10 degrees in an exopolymer-rich solution. We've also demonstrated this in the field using a digital holographic microscope, but I don't have time to show you that. So we've emerged from years of work with this image of what's going on in a sea ice pore, the microbial ecosystem that lives there. And this is all based on quantitative data. So you see lots of viruses in here. And we had the question, do exopolymers protect against viral infection? In the virology world, there's this war terminology about winners and losers, those that infect and kill and lyse. And so we were in that mode. Do exopolymers protect against this? We don't have a totally clear answer to that, except that the high numbers of viruses do suggest production of viruses. Though, again, Max's poster this evening can tell you more eloquently about that. The more important or profound question I've come to understand is, do microbes benefit from gene exchange that they experience as a result of interaction with viruses? And can this account for their adaptability to these extreme conditions? So we have a new project, or we're in the midst of it, supported by the Moore Foundation, to study gene flow in Subzero brines. And we're comparing gene flow in sea ice, a modern contemporary environment that I've already talked with you about, and cryopegs, which represent ancient brines. So we can access both of these environments from Nukjavik, Alaska. We can go out onto the frozen ocean for the sea ice, and going too fast here, we can access cryopegs through this permafrost tunnel, which is just a box in the middle of a frozen field. And when we first opened it up to see what we were encountering, is a little bit daunting as well as exciting. If you've been in an alvin dive before, this is like diving into the frozen bowels of the earth. So we had to climb down into this tunnel, which was excavated from ice and permafrost. So it's 6 meters or so deep, tunnel in here, and through boreholes from the floor of the tunnel, we can access marine sediments that are saturated with salt, with liquid brine. And this is what's called cryopeg by the Russians, who have first studied these cryopeg brines. So the brine is at -6 to -8 degrees, 115 to 140 parts per thousand. So I think that's an extreme environment. And these are ancient relic seawater brines. The permafrost around here is dated at about 50,000 years. C14 dating puts a minimum at 11,000 years. So we'll call this ancient seawater brine. The Russians have already projected this as an analog for the Mars subsurface, so we're thinking along those lines as well. Here we are in our frozen cave, and when we finally collected this golden solution from below the floor, we had some surprises in store for us. We actually have an organic soup under these very extreme conditions. Here is plotted dissolved organic matter, which is orders of magnitude higher than anything in sea ice or seawater. Most of it is accounted for as exopolymeric material. When we just put a drop of this liquid under the microscope, it's jam-packed with microorganisms, 10 to the 8 per mil. We were totally surprised by this. So now we're launched into a complete omics analysis of what we found. So we don't have the low biomass problem here. However, the brine has presented us with some extraction problems. But in any case, we're moving forward with our omics discussions here. How am I doing? 2 minutes, okay, to get to the heart of the matter here. Okay, so Josie Rapp is my postdoc who's not here right now, but she's put together a comparative slide here of cryopeg brine and sea ice brine. These are all the functional gene categories that exist in our current databases. To tell you all the different functions of cellular life, of microbial cellular life. I just want to draw your attention to defense mechanisms or evidence of phage-host interactions. So in the cryopeg brine, we have a greater relative abundance of those genes than we do in the sea ice brines. This was unexpected because sea ice is a fluctuating environment. I was expecting more interactions with viruses there than in the ancient cryopeg brines, which have been stable, geologically isolated in the dark for 50,000 years. Instead, we have more evidence of more viral interaction. And this is just a visual depiction to convince you, showing you all of the genes, that the cryopeg brine is very different from the sea ice brine. If I just pull out, or if Josie just pulls out, a set of genes that indicate phage-host interaction, CRISPR genes, I'm sure some of you are aware of these, you can see the higher abundance in the cryopeg versus sea ice. And just to pull out one example, it's orders of magnitude higher. And here is a co-assembly of the cryopeg brine samples for genome binning. So if you're not familiar with these, the thing to realize is that these gray layers here are our cryopeg brine samples. These are all the genes that were present in those brines. Here are the bacterial hosts that we can identify. And importantly, here are the viral genes that we're picking up throughout these metagenomes. These viral signals, 927 of these pieces of DNA, these contigs, which corresponds to over 20,000 gene calls for viruses in the existing databases. So we're not accounting for anything unknown yet, or if we can ever. And of these, 915 correspond to auxiliary metabolic genes, AMGs. So if you're not familiar with those are genes delivered by viruses to the host cell that improve the condition of the host cell, its metabolism. So here is clear evidence that viruses are helping these microbes to succeed in their environments, or I would suggest it's clear evidence. And just zooming in a little closer here, these are the viral contigs here, which we can link to their microbial hosts so that we can put together specific phage-host interactions that have happened, gene transfers that have happened, if not favored, have enabled life in these extreme environments. Okay, want me to zoom forward here and conclude. We have evidence of viral AMGs for exopolymer production, for cryotolerance, osmotolerance, membrane fluidity, so we're getting very close to supporting our hypothesis. And I would just end by saying that as we search for microbial life as we know it, what we're really searching for is this co-evolving pair or sets on the spectrum of life between microbes and viruses, which is going to complicate our search but make it more realistic. Thank you.
All right, thank you, Jody. So let's start off with a couple of quick questions for Jody. And by the way, for anybody who just came in here, we're going to spend the next 15 minutes as kind of a break time. So we'll have questions for Jody and also for Rebecca for the first talk of the session.
Yeah, I'm Charlie Lineweaver from the Australian National University. It seems to me that the environment that you're investigating here is one of the few places on Earth where you have, I've been told by many people that the ice caps go away. And for most of the life of the Earth, they haven't had ice caps. So you're in an environment which you have ice caps and you don't, you have and you don't. I would have thought that would, you talked about cryotolerance and the genes for cryotolerance. I'm not sure how soon we will lose our ice caps, but when we do, will all the organisms that you're studying go extinct, or will they somehow try to adapt to go further south, and then they will have to re-evolve cryotolerance genes that you're analyzing here when we get another ice cap, et cetera, for thousands and thousands of times?
Yeah, no, I understand your question. If you all didn't get it, it's how do these cold-adapted microbes survive between periods when we don't have ice on the planet? So we don't have a clear answer on that, but my view is that microorganisms retain the memories, the genetic memories of where they have been before for very long periods of time. As long as there's some minimally cold environmental refuge for them on the planet, it could be in the deep sea at 15 degrees, it doesn't have to be frozen. They can retain the genetic information they need. And if they don't retain it, the pool of the pan-viral genome will retain it for them.
So you see evidence of cryo-tolerant genes that are older than multiple episodes of ice caps going away, coming back, going away, coming back.
Great question.
Okay.
We'll look at that.
I was going to ask something similar. Can you see differences in the mutation rate or the sequences of some of the auxiliary metabolic? And then a related question would be, I've just heard of CRISPR in terms of gene editing, and the insertion of a CRISPR gene itself, does it alter the flanking sequences that will ultimately change the nucleotide sequences within the gene itself? Can you, in other words, can you sequence these genes and see how they're evolving between the same?
Yes, so I've now gotten involved in your second question mentally. What was the first one just again?
Are the genes themselves evolving? Can you see differences between
okay, so I don't have an answer for you, but we definitely intend to look at that using whatever tools we have. There'll be bioinformatic tools where we have to make assumptions about evolutionary rates, but we'll definitely be looking at that.
So there are a number of ways that they could be evolving.
There are a number of theoretical ways to look at that. Yeah.
I have two questions. One is, you alluded to it but never said it, I think, and that is that viruses play a role in actually turning on the genes for the extracellular polymer. Is that correct?
So I didn't get to show you that last slide very well. All we've done so far, mind you, we just received the sequences, so we've pulled out this set of 12 genes that are associated with exopolymer production, and they are virally associated genes. They are genes that are recorded in databases as having come from viruses. So the actual regulatory mechanism I can't comment on with the metagenomics, but we also have metatranscriptomic data, which we've not examined yet. So you have to stay tuned for that, sorry.
The second question is the EPS. Is an appreciable amount of that a polysaccharide? And if so, are the dominant sugars uronic acids like black uronic acid and glucuronic acid like you find in other organisms, other marine organisms like cyanobacteria?
I can't answer that question because the method we're using for extracting is the phenol sulfuric acid method. If we were using an Alcian blue stain, I could tell you yes. I believe that they're similar. They behave like our sea ice polymers do. So I believe they're similar, but we haven't done the chemical analysis.
Carl Pilcher, Blue Marble Space Institute of Science. Jody, great talk.
Thank you.
I don't have a question, but I just had to make the comment that Barry Blumberg would really, really have liked this talk because, as you know, Barry emphasized that life is about cooperation more than it is about competition, and you've just given us a beautiful example of that.
Thank you for that. I do enjoy thinking about viral host interactions as altruism and not in the war terminology that infiltrates our literature. I do believe that they are helping each other as they co-evolve. It's all part of one collaborative evolutionary effort.
So I actually have two questions. One, I am actually wondering if you have looked at plasmids at all in terms of conjugation and transformation as potentially being related to any of the HTT that's present in these samples. I'm actually with the NASA Gene Lab developing a pipeline for performing first computationally deriving plasmid sequences from metagenomics data and performing gene ontology enrichment analysis. So I would be happy to potentially collaborate with you if you were interested. Coincidentally, I'm also in the market for a PhD program. But
incidentally, I'm near retirement. As far as I know, we haven't looked at the plasmid question. Can anybody from my lab elaborate on this? I don't think so. I don't think we've looked at plasmids yet.
We haven't looked at plasmids specifically yet, but I mean, in the whole suite of all of the gene transformations that we want to look at in our metagenomes, it's certainly occurring. That along with conjugation methods and just any natural competency evidence that we can find. So that we know that there's a variety of processes that can dynamic.
We have a whole list of questions that we can now address now that we're sitting on all this omics data, and we're just at the start of it, so forgive us that we haven't got all your answers.
Okay, and the second question was, so I saw that a lot of the genes that came up were CRISPR associated, and I've heard that it just, I guess in extreme environments you tend to have very streamlined and very confined genetic suites. So I'm wondering if, what you're seeing is you're seeing CRISPR association implying microbial viral resistance genes, right? But also seeing these metabolic genes. So it's almost like...
Right. So it's a surprise, isn't it? I had gone into this thinking that at the low end of the temperature range, we would see fewer and fewer CRISPRs. There's some literature that suggests that. And so we haven't completed our comparative analyses of how many CRISPRs in these extreme environments compared to other environments, but it's looking surprisingly high. I don't know how to explain that, except that extreme environments are always presenting us with surprises, and we need to keep an open mind about our mindsets.
This is really high. So that was a really great talk.
Thank you.
You just said you didn't really like talking about the competition, but my question's kind of involving what, if you could comment sort of on the competition, because I thought that that's a shockingly really high biomass environment. Yeah. And so maybe you probably haven't looked at any of these antimicrobial compounds or genes or anything, but if you were to kind of speculate, what could you speculate about that?
You're right, we haven't looked at those things yet. We have other measures of activity going on in these samples. We have measures of high extracellular enzyme activity, or high for the conditions that they're under. We have evidence of dividing cells. We have evidence from BondCat, which is a relatively new method that tells you that they're consuming amino acids. We don't have anything that I can use to comment on the competition question other than the bacteria to virus ratio, which is surprisingly low. It's on the order of 1 or less in the cryopeg brines. It's higher in the sea ice brines, which is why I was expecting to see more infection and more CRISPRs and more interaction at the competitive level there, at the lysis level.
How would that compare to like just outside soil environment?
Beats me, we'll have to look at that.
Thank you.
I'm an oceanographer.
Thank you for a really wonderful talk, Frank Rosenzweig, Georgia Tech. A couple of related questions, and questions that sort of feedback onto an early question about mutation rate, and that is, what handle do you have on generation time in the sea ice versus the cryopegs? Mm-hmm. And then related to that question is, what are the sources of primary productivity, and is that Is that continuous and slow, or is it episodic? And again, if you could contrast those two environments, that would be great.
So in sea ice, it depends on where you are in the ice, but if you're at the bottom of the ice where light penetrates and nutrients are from below, you have plenty of primary production through ice algae, which Jody Young will talk about shortly. In the cryopeg, it's totally dark and has been totally dark for 50,000 years. We have some evidence, I don't know if she's going to talk about this or not, of rubisco genes in there that could be utilized by chemoautotrophic bacteria. But they would be very rare in occurrence because our data is telling us that this swamp of bacteria are heterotrophic bacteria. So it is a puzzle as to where all the carbon is coming from. And we are definitely puzzling over that, trying to get datings on things and sources of organics here, and it's a difficult question. And I think I've probably forgotten your first question.
No, that satisfies me.
Yeah,
it's really
okay.
a wonderful system, so
Okay,
thanks
thank
a lot.
you. It's a wonderful system, but there are a lot of unknowns about it and a lot of things we have to wrestle with.
Hi, Jody. Sorry, yeah, I guess I'll be the last question.
Yes, and he's the one that invited me to do this, so thank you.
Thank you so much for coming out. It was a great talk. It's exciting. For the AMGs, they can be hard to discover, and now you guys are doing more work. Were there other AMGs that you didn't talk about for the sake of time? Did you find anything for antifreeze proteins or anything else like that?
Yeah.
Oh,
Yeah, I haven't listed them out here,
well,
but...
so yeah, sorry.
but
I was thinking these were all...
we have comparable tables for cryotolerance, osmotolerance, membrane fluidity. Out of Matt Sullivan's lab, Jiping Zhang, his postdoc, has identified a lipid desaturase, which keeps the membrane fluid under very extreme cold conditions. That is an AMG, virally transferred. He's identified that in the viromes from the cryopegs. So yes, we have some very clear evidence there.
I also wondered, there's this idea of this arbitrium system, where it's virus communication via adapting the host quorum sensing. If you think maybe here they're producing this, they're helping the host produce this EPS, and maybe they're communicating to have these guided decisions of when to burst out of the host and when not to, and that might affect the virus host abundance.
Very cool idea, yeah. We haven't measured quorum sensing compounds here. We'd be happy to give her a sample.
Thank you so much.
Yeah. Thank you.
Let's thank our speakers once more.